I'm going to present the results of 2 studies which are fairly recent, published in the last 2 years or so, but I'm going to put them within the context of some much longer-term trials that we've been doing looking at phenolics, and the reason that I'll be doing that should become clear as we go through. Perfect. So at Northumbria University, I manage an interdisciplinary research group called NUTRAN. And essentially, our mission is to improve health and well-being through nutrition. And that can mean for some people via foods, so whole foods, like the Mediterranean diet, for instance. But for us, that usually means supplementation, which for some people can be a bit of a dirty word, especially in the academic world. You know, ideally, we'd love to get all of our nutrition from the whole foods that we eat and for them to be the best foods that they can be. But for lots of people, that's not possible. And so we've been looking at the effects of nutritional supplementation for the last 15 years or so within our university. And over the last 2 years, I've been doing that whilst managing this group called NUTRAN. And our group really brings together the world-leading expertise from the Brain Lab, which has been going for, as I said, over 15 years now under the directorship of David Kennedy. But now it also encompasses the biological lab, so our ability to measure all of the wet stuff that we can take out of people, so stool, urine, saliva, blood. We can measure those samples for a range of biomarkers, whether that be stress, inflammation, oxidation, measuring the levels of the intervention itself, so measuring polyphenols in the blood, or a whole host of other interesting markers. We also have the sports lab as part of NUTRAN, which looks at physical performance effects of nutritional supplementation. The sleep lab, so a purpose-built sleep center with 2 bedrooms where we can monitor the effects of nutrition on sleep. The vascular lab, which looks at blood flow in the periphery, so in the body. And the omics lab, which looks at the gut bacterial community. So we have some state-of-the-art sequencing facilities to be able to look at what's happening in the gut. And I sort of sit in the middle of all of those colorful blobs, trying to get everybody to work together to do more interdisciplinary nutrition research trials. So not necessarily just looking at the brain and or sport, but can we get more of those labs working together to create much bigger, much more complex, but ultimately much more interesting research trials to answer the more complicated questions which exist nowadays. I've kind of covered that, so I'll skip on to the more interesting stuff. So, as I mentioned before, we've been looking at polyphenols since about 2008, which was when I started my PhD at Northumbria, looking at resveratrol. And I've highlighted in bold the studies that I'm going to talk out, that I've kind of picked out to talk about today. So I'll start by talking about those historic trials with resveratrol, and then I'll move on to those more recent studies looking at more complexes of polyphenols in humans. So, a very busy slide for this next slide, so apologies for that. But essentially what you're seeing on the screen in front of you are some graphs which we've taken from the neuroimaging studies that we've done with resveratrol in the past. So this is a type of neuroimaging called near-infrared spectroscopy, and it measures blood flow in the prefrontal cortex. And all participants need to do is wear a headband across the forehead, and they can engage with their environment. They can sit and do cognitive tasks on the laptop. They can move around and engage. They don't need to be sort of static and still in an fMRI, for example. So it's a really valuable neuroimaging technique for this field of research. So all 3 graphs are from that near-infrared spectroscopy looking at blood flow. And essentially the message of this slide is that resveratrol really does improve blood flow quite consistently and quite robustly. So all of those lines on the graph, the ones at the top, the ones with all of the lovely stars, they show significant boosts in cerebral blood flow after participants have had resveratrol, which for those of you who don't know, it's a polyphenol largely present in grapes and red wine. Hence a lot of us are much more interested in it than perhaps other polyphenols. So, a really consistent finding, but one thing that was also consistent is that improving blood flow in the brain does not improve cognitive function. So just by virtue of giving people access to more blood, more oxygen and glucose in that blood, doesn't mean that that brain region works better. and it doesn't mean that you see improvements in performance as a result. It took about 10 years to answer that question during a PhD, where you make sort of small changes and you try and do things better and differently, but ultimately it just doesn't. And that kind of led us to think, well, is it because it's a single polyphenol? Is it just not active enough? Even though it has these robust mechanisms of action, maybe they aren't sufficient to improve cognitive function. So what if we give people polyphenols in complexes, so more than one at a time? Would they be more likely to improve cognitive function? So if we look at this first study, which was published in 2020, but this is actually the first time it's been disseminated in any kind of conference environment. So in this study, all participants, so all treatment groups, were based on a base drink. And this drink contained beetroot, which has got lots of dietary nitrate and betalains in, and ginseng, which obviously has ginsenosides. It also had some sage in there as well. And that was our sort of botanical base drink, to which either blueberry, apple, or coffee berry was added. So all had the base, and then they had these additional compounds added to them. So either rich in anthocyanins, catechin polyphenols, or chlorogenic acids. Fairly similar to the previous trials in terms of the number of participants, in terms of the types of cognitive tasks that we were doing, which are just on the bottom of the screen there. So otherwise very similar. The only difference is there's lots more going on in terms of supplementation. And we saw some really lovely effects. So in terms of blood flow, these polyphenols also boosted blood flow in the prefrontal cortex. So we saw that with levels of total hemoglobin, oxygen saturation, rising as a result, in particular, of the apple and the coffee berry. In terms of cognitive function, all drinks boosted some aspect of cognitive function. So the blueberry and apple had effects on anger, hostility, and depression, dejection. So lowering those or having a more positive effect on those. And then the coffee berry also had positive effects on confusion, bewilderment, and fatigue and inertia, as well as total mood disturbance. And this is from a questionnaire which assesses lots of facets of mood. And finally, apple and coffee berry increased alertness and reduced mental fatigue in participants. And that's why those two interventions in particular, coffee berry and apple, were taken forward into a second study. So this was published a year later, in 2021. A slightly higher sample size, and the reason for that is that we didn't measure blood flow in the brain this time round. Our assumption was that we'd already shown that in the study the previous year, so we didn't need to again measure cerebral blood flow. But what it also meant is you can then increase your sample size. So one of the restrictions with neuroimaging is you can only put it on one head at a time, and that really limits the number of participants you can have in your lab assessing their cognitive function. If you're just doing the latter, you can have quite a large testing lab with maybe 10 or 15 participants assessing their cognitive function at the same time. So it hugely frees up the time and space that you have to measure cognitive function. So this study, the base drink, was a much more inert base drink. So this was just water with sort of taste additives included. And that was really just to mask the taste in all of the conditions. So the drink tasted the same in each of those. And then participants either had coffee berry at 1.1 grams, which was the same in the study previously, or they had that with the apple extract added to it, or they had a low-dose coffee berry, so just 100 milligrams with that apple extract added to it. Again, the cognitive tasks were the same. The only difference here is that we didn't measure cerebral blood flow at the same time. And what we showed was a really nice reproduction of what we'd shown previously, which is the holy grail in any type of research, that if you're doing something fairly similar, over time, you'd like to be able to show the same pattern of effects, which is what we showed here. And in red are those findings which were exactly the same as the previous study, which is most of them. So coffee berry on its own showed positive effects on alertness, higher vigor and activity, and these are all subjective reports from our participants, with lower fatigue inertia, and again, lower total mood disturbance. So again, really similar replication of what we'd shown the year before. Additionally, we found some more nuanced cognitive effects. So RVIP is a task which requires quite consistent engagement for about 5 minutes. On the screen in front of participants, you have numbers flashing up at about that rate. And when participants have seen 3 odd or even digits in a row, they need to respond. So 5 minutes is a really long time to do that, and what we see is that performance dips as a result. People get tired, they disengage from the task. And with this study, that was around the 3rd repetition of doing this task, but what Coffee Berry seemed to do was to ameliorate that. So participants got less fatigued, disengaged less with the task if they'd had the Coffee Berry. But conversely, this did lead to higher false alarms. So the number of times participants were responding and there weren't 3 odd or even digits in a row. So there was a bit of a trade-off there in terms of accuracy and speed. And finally, Coffee Berry at the higher dose with apple resulted in faster completion time for what is quite a traditional task, I would say, in psychology. So peg and ball is sometimes also referred to as the Tower of London task. It requires participants to look at a goal configuration and then to try and copy that by moving pegs around on balls. So it takes a lot of thinking time, but then it also takes completion time to do that. And we found that Coffee Berry improved the latter. And I guess what we're seeing there is probably Coffee Berry is improving alertness, and that increase in alertness is having all of these additional positive effects on other areas of cognition. So if we go back to the previous table that I showed earlier on in my slides, what we see is those earlier studies that we did with phenolics, where we were administering single compounds, so resveratrol, which we've done most of our research on, but also green tea, which we looked at quite a long time ago now. They have effects elsewhere, but what we tend not to find are very robust effects on cognition. If they do exist, they tend to be fairly weak. But all of the studies that we've done subsequently, which either give multiples of polyphenols or maybe in addition to other phytochemicals, phytonutrients, those are the ones which also produce those effects on cognitive function. And I've talked about the ones in bold today, but there are many more which also kind of follow that same story, which is really interesting. Now, it might be that they are as a result of, well, that's how we consume polyphenols in nature. We don't isolate individual compounds and consume those. And so it sort of stands to reason if there is an effect which is produced in nature, that you would see the effect when you consume that compound in its natural kind of state. You've got all of those sort of symbiotic interactions occurring when you consume them. It might also be a methodological issue. So I mentioned previously that when you're doing studies looking at neuroimaging, you're really restricted in terms of your sample size. So it may be that a lot of those cognitive studies were really stymied by not having sufficient power to find those cognitive effects. However, that doesn't explain the first Coffee Berry study, which only had 32 participants, which is considered fairly small in terms of cognitive nutrition research. So that argument doesn't necessarily hold true for all of these studies. So yes, but that's certainly something that we consider now in our research centre. And oftentimes now when we're designing trials which are looking at both cerebral blood flow and cognition, what we tend to do now is have the cognitive side adequately powered, so that's sufficiently powered for cognitive function, and then we only measure cerebral blood flow in this case in a subsample of those participants, because you don't need as much power for those analyses. So that's why those figures tend to be smaller for the cerebral blood flow. And I think that's it. Fairly short and sweet. Lots of time for questions if anybody has any.
Thank you so much, Dr. Whiteman. So, yeah, let me bring the mic over to you if you have a question. And I also just want to remind people that we are filming this and we're recording also. So I know that sometimes people take photos of slides, and those slides will be viewable on Vitafoods Live as well.
So, Dr. Mark Miller, couple of questions, and we'll try and layer it out a bit. I assume most of the polyphenol work initially was trying to enhance, with the focus of blood flow, nitric oxide mechanisms based on limiting superoxide destruction of nitric oxide, which can occur instantly in essence. But so, and that's why you can see acute changes in blood flow.
Yeah.
I think design-wise, though, there are better questions to ask with a change. First of all, you had a fairly young population, which I presume were university students.
Yes.
Yeah. And they really are not suffering, or at least don't have a problem that's associated with aging. So if you had an older population where there may be either compromises in blood flow or certainly some detriment as it relates to cognitive function, and then also don't do an acute event, because you're not really worried about these, well, I'm not as worried about the superoxide, nitric oxide acute events, because that may be short-lived and may not translate. And in the elderly population, it's not only just blood flow, it's really glia and other supportive cells that have macrophage-like function that's really driving neurocognitive issues. So I would suggest that an interesting design would be not do it acutely, do a subacute or a chronic intervention with whatever combination you want, and do it in an older population. And would that begin to tease apart the types of answers that you may be looking for?
Absolutely. Yeah, I think most of us in the nutrition world know now that either a lot of these interventions just aren't effective in the young, healthy populations, and or they're probably not beneficial either. And so why investigate these groups? But it can often be a really good first step, as you said, being on quite a big campus. They're relatively easy participants to access. And so answering those initial questions, like with resveratrol, for instance, you know, they're fairly low-hanging fruit to investigate, but as you say, definitely need to be working up towards those older age groups. We tried a few years ago to inhibit cognitive function in those young, healthy samples as a bit of a bridge to get to those older groups, which are quite difficult to access as cohorts. So at Northumbria, we've got an environmental chamber which can mimic altitude, you can change the temperature. A lot of athletes come to kind of train in that chamber for performance. And what we try to do is take young people to a high altitude, about 4,000 meters above sea level, and we definitely inhibited their cognitive function, but resveratrol didn't rectify it. Again, it might just be that those effects are too acute, and you know, we just weren't observing them. But yeah, I think that taught us that probably skip that model stage and just go straight to those older participants. And the only issue there is just funding. Nobody necessarily coming forth with the funding for those types of studies. So that's often the limiting factor as well, unfortunately.
Funding is always a limiting factor. And I was going to ask you relatedly if there was a plan to do studies for older populations. And another interesting model would be to look at a macro population study and look over time. Yep. So if you're looking at young populations and some of those, perhaps they're acute, but perhaps there's the longevity effects. And if you had controlled populations, that would be of interest. But are there any plans, funding notwithstanding?
Not in the immediate pipeline. I guess the SAGE study, which was on the slides, not in bold. I didn't talk about that one today. That was in a slightly older population. That was a study funded by Nexera that we published a couple of years ago, which, again, we saw some really nice findings there on cognition, potentially because we were moving into that older sample. And then we've got a study which is in prep at the minute, which was funded by Puka Herbs, who are a UK-based company. Again, moving into older populations, but not that elderly group, sort of sub-elderly, I guess.
All right, any other questions in the audience here? I'm also checking online. I don't see questions coming in online. Open. And is there anything else you'd like to add?
I guess just watch this space. So I know a lot of, so David and I, who's sat in the second row there, you can give people away. David's the director of the Brain Lab, who's part of NUTRAN. David's been doing this for way longer than I have and working with industry to produce these findings, to make claims. I guess sometimes for very small companies or very new companies, it can be quite a scary arena to try and get into, you know, engaging with research. I think sometimes engaging with a university for research is sometimes a bit less scary, maybe a bit more engaging. So if anybody is interested in talking about research opportunities, then you have my email address there. Just get in touch directly. You know, we have companies who come and they've just have a product. They don't really have ideas yet, and we're happy to sort of talk through all the way from that stage through to people who say, this is exactly what I want. This is what I want investigated, and we do that. We do that research. So just get in touch if anyone's interested in exploring that further.